Good morning to everyone. Thank you very much for me to be here today. We will have Tânia Dias. Tânia Dias, I hope that all of you already know her, but if not, Tânia Dias is a researcher in one of the projects that we have ongoing right now. So a project which is devoted to the study and valorization of cyanobacteria. This project is being developed together between us at PATH and Joana Pereira and Professor Fernando Gonçalves from the biology department that you already know. So with further delays, my pleasure to introduce you Tânia Dias, and Tânia, the stage is yours.
Good morning, everyone. Probably this is the first time that many of you can see me without a mask due to the current world situation. So today I will present the work I developed during my PhD. But first I will just make a brief overview of my academic path so you can understand how I got here. So I completed my degree and master degree in biomedical sciences at the University of Beira Interior in Covilhã. And during my master, I started working in the field of male fertility and diabetes, and I had the opportunity to continue in the same field for the PhD. So in 2012, I enrolled in the PhD in biomedicine at the same university and in the same research group. And it was the first year I applied for an FCT fellowship, and I did not get funded. So in 2013, I got a 2-year CRAN fellowship within a project called Hormonal and Inflammatory Bases of Age-Related Diseases. And I developed this project together with my PhD research plan. So I continue applying every year for the FCT PhD fellowship. And only after applying 4 times, I got the fellowship in 2015 in the field of medicinal chemistry. But it was not that bad to not having the fellowship right away, because it allowed me to collaborate as invited assistant professor in several classes related to human genetics for 5 academic years. At that time, I was teaching about 32 years per academic year, and this reduced my tuition fees while I didn't have the fellowship. So by the same time, my research group had to move to ICBAS in Porto, to the Institute of Biomedical Sciences, Bel Salazar. And I also moved with them because part of my PhD was supposed to be developed there. I stayed about 2 years at Porto, in Porto. And then in 2017, my PhD project was almost completed, and I still have 2 years of fellowship. So I wanted to have an experience in a foreign country, and I decided to apply to a Fulbright research scholarship with FCT support. This was the first time that FCT allowed the accumulation of both fellowships. So I went to Cleveland, Ohio, United States, to work as a visiting researcher in the American Center for Reproductive Medicine. And I was working in a project called Understanding the Molecular Mechanisms of Male Infertility in Men with Testicular Cancer, utilizing proteomic tools. Although this project was also in the field of my PhD project, it was not included in the thesis. So I came back in May 2018. And for 1 year, I was just finishing writing papers and writing my PhD thesis. I completed my PhD in 2019. That I will present after this. And for 8 years, I was working in the same field and doing the same techniques, and I was proud of my work. But I could not see a real life application of what I was doing because it was basic research. So I started applying to different fields and to projects in which I could see the translation from the bench to the real life application. And this is mostly how I got here. As Sonia explained, I am now a junior researcher in this project, Refined Cyano. So I had a 6-year gap between my PhD and this position. So my PhD project was in the field of male fertility, and it was a little bit different from what is done here. So I will not get into much detail, but I will make a brief overview. This project was entitled Effect of YT on the Reproductive Function of Diabetic or Prediabetic Individuals. And why did we study diabetes? Diabetes is a chronic disease characterized by hyperglycemia, and its incidence is increasing over the years worldwide, with 425 million cases in 2017. In that same year, it was the direct cause of death of about 4 million people. And it is expected that there will be an increase of about 200 million new cases by the year 2045. So there are two main types of diabetes. The type 1, which accounts for about 5 to 10%, is characterized by a deficient insulin production. And type 2, which is the most common type, is characterized by an inability to use insulin, also known as insulin resistance. There is also an intermediate state known as prediabetes, that in which the glucose levels are higher than normal, but not high enough to be considered diabetes. So, this condition can occur up to 10 years before the progression to the type 2 diabetes. And this is why it is important to study it, because it can still be reversed with lifestyle changes. In 2017, about 352 million people were diagnosed with prediabetes. However, most of the cases are still undiagnosed, and people only go to the hospital when they have symptoms, and this is why it is not diagnosed. Diabetes has a very negative impact on male fertility, with 50% of male diabetic patients having some degree of subfertility. In particular, 35% of the male patients with type 2 diabetes are infertile. And these figures are quite worrying because they are increasing in men during the reproductive age. Among the reproductive alterations, we can the most severe are the alterations in the metabolism of reproductive cells, increased oxidative stress that can lead to damage to DNA, proteins, and lipids, and altered spermatogenesis due to hormonal dysregulation and altered Sertoli cells function. Ultimately, this will result in decreased sperm quality. These changes are further aggravated by the lifestyle factors, such as poor eating habits or physical inactivity and environmental factors. Thus, one of the primary strategies to treat diabetes and its complications includes nutritional changes and physical activity, which are usually complemented with anti-diabetic drugs. However, the existing drugs so far have still limited efficacy and long-term costs and secondary effects. Recently, natural products obtained through the diet demonstrated interesting antioxidant and anti-diabetic properties, including tea and its constituents. Tea is one of the most consumed beverages worldwide, and it is obtained from the plant Camellia sinensis. And depending on the processing of the leaves, we can obtain different types of tea: white, green, oolong, or black. White and green are the least processed types, and then they retain a higher antioxidant potential. We decided to study white tea, not only because it usually has a higher antioxidant potential, but it was also the less studied. At that time, there were only a few publications about white tea, and now there are about 100 per year. So the objective of this project was to study the effects of white tea on male reproductive function and its possible role its possible protective role against reproductive dysfunction induced by prediabetes. However, besides, we also want to investigate if the effects of white tea were due to a combined effect of all its components, or if there was a stronger effect of one of its most bioactive components. So we started by analyzing the white tea phytochemical profile, and then the project was divided in 3 parts: the in vitro studies using rat and human Sertoli cells, the ex vivo studies using rat spermatozoa, and the in vivo studies in which we developed a prediabetic rat model. The phytochemical profile of white tea was evaluated by proton nuclear magnetic resonance, and through the identified compounds, we selected the most representative compounds from each family, from the methylxanthines family, amino acids, and phenolic compounds. Caffeine was the most abundant methylxanthine, and it is responsible with the stimulating properties of T. The most abundant amino acid was L-theanine, that is almost exclusively found in the tea leaves, and it is responsible for the relaxing properties of tea. Among the phenolic compounds, we selected the pig-allocatechin gallate because it was the most abundant and to which most of the benefits of tea are attributed. So, starting by the in vitro studies, we evaluated the effects of an aqueous extract of white tea and the 3 selected compounds individually in Sertoli cells. And these cells are important because they are the functional units of the seminiferous tubules of the testes, here represented in yellow. They provide the physical and nutritional support to the developing germ cells, since the undifferentiated cells close to the basement membrane, that are called spermatogonia, until the release of the fully differentiated spermatozoa in the lumen of the seminiferous tubules. Adjacent Sertoli cells form a barrier that is called the blood testis barrier and divide this tubule into basal and abdominal compartment. All the cells included in the abdominal compartment are dependent on the products resulting from the Sertoli cells metabolism. This is a representative figure of the Sertoli cells metabolism, in which the preferred substrate is glucose, and it enters the cells through specific transporters that are called glucose transporters 1, 2, and 3. And the glucose is converted to pyruvate through glycolysis, and then pyruvate can follow 3 different pathways. It can enter the mitochondria and form acetyl-CoA and fuel the Krebs cycle, and it can also be converted to alanine, which is involved in maintaining the redox state and glucose homeostasis. And it can also be converted to lactate by the enzyme lactate dehydrogenase. And this is the preferred pathway of Sertoli cells metabolism. They function a little bit like cancer cells. Because lactate is a metabolite that is very important for germ cells, because some of the stages cannot use glucose. So they need lactate for their energy supply. In the in vitro studies, we used primary cultures of rat Sertoli cells and also human Sertoli cells, because at that time, when we started the study, we didn't have access to human Sertoli cells, so we started with rat primary cultures. And we exposed cells to either to the white tea extract or to the compounds individually at different concentrations. And after 24 hours of exposure, the culture media were collected to evaluate the production and the consumption of metabolites by proton nuclear magnetic resonance, and the cells were used for the evaluation of viability, proliferation, and also expression levels of several intermediates of glucose metabolism, mitochondrial function, and oxidative stress. Regarding the study with the white tea extract, it was observed that cells supplemented with white tea consumed less glucose. And this was mediated by a decrease in some of the transporters and also the enzyme phosphofructokinase 1 that converts glucose to pyruvate. However, there was an increase in the enzyme lactate dehydrogenase, and this resulted in an increase in lactate production, which could be important as lactate is used as energy source, as I said before, but it also has an anti-apoptotic effect on these cells. In the case of caffeine, although there were no changes in glucose consumption, there were several metabolic adaptations that resulted in a higher lactate production at the lower concentrations. However, there was a dose-dependent decrease in the antioxidant potential, which led to an increase in oxidative damage at protein level. So it demonstrates that it can be dangerous to a higher consumption of caffeine. In the case of the phenolic compound, the pig-allocatechin gallate, there was an increase in glucose metabolism in the cell supplemented with the highest dose. However, the metabolic adaptations resulted in the maintenance of lactate at normal levels. There was also a decrease in the lipid peroxidation in the same group of cells, which was associated with the higher potential, with the high antioxidant potential of this phenolic compound. In the case of the amino acid L-theanine, this was the compound with the least impact on Sertoli cells metabolism. It induced an increase in glucose consumption at the highest dose and maintained lactate at normal levels. So with these in vitro studies, we observed that both white tea extract and the components individually can regulate Sertoli cells metabolism while maintaining or increasing the production of the most important metabolite for male reproductive potential, which is lactate. However, it is not clear if white tea is better for Sertoli cells function or the compound individually. So we proceeded to the ex vivo studies with all the groups. In this type of studies, we evaluated the effect of white tea and the selected compounds individually or in combination when the spermatozoa were stored at room temperature for a short period. Sperm quality is an indicator of male fertility potential, and over the years, many studies reported deterioration in sperm quality. Sperm viability decreases dramatically when samples are stored in refrigerated environments due to the increased production of reactive oxygen species over time. So this is one of the parameters that are mostly evaluated. And the short-term storage at room temperature can be an effective alternative for the transport of samples, for example, or also during assisted reproductive techniques, in which it can be beneficial to incubate the samples at room temperature, avoiding refrigeration, which will lead to higher reactive oxygen species formation. So in these studies, we used spermatozoa from Wistar rats with 3 months old, 3 months of age, and they were incubated either with the white tea extract or the constituents individually. In this study, we also compared the white tea extract and the green tea extract. And during 3 days, every 24 hours, we evaluated the antioxidant potential of the cells, lipid peroxidation, and viability. Here, I will present in these graphs, the blue will be the control, the green is the green tea, and the yellow the white tea. So what we observed was that the white tea extract induced the highest antioxidant potential in the spermatozoa, a big difference for the control and the green tea. Relative to the lipid peroxidation, white tea supplementation reduced significantly the oxidation to lipids during the 3 days of storage at room temperature. And in terms of sperm viability, white tea supplementation kept spermatozoa with a high viability compared to the collection time, which was measured after the sacrifice of the animals, and it showed better results compared to any of the compounds individually. So with these studies, we concluded that white tea extract can be a cost-effective additive for sperm storage media, facilitating transport at room temperature and avoiding deleterious effects of refrigeration. These studies highlight the importance of the combined effects of all the white tea components rather than using the compounds individually. So in the in vivo studies, we proceeded to the evaluation of the effect of white tea in reproductive tissues, namely testes and epididymis, and we also evaluated the sperm quality in an animal model of prediabetes. So we used 18 Wistar rats, male Wistar rats, and at 2 days of age, they were divided into groups, and the 6 groups in the control and 12 groups to induce the prediabetic state. This was induced with an injection of streptozotocin. And after 1 month, one of the prediabetic group was divided in 2. One group drinking water, and the other one, the water was replaced by white tea. So they were drinking white tea for 2 months. At 3 months of age, glucose and insulin tolerance tests were performed to evaluate if the animals developed the prediabetic state. And then they were sacrificed to collect the reproductive tissues and evaluate the metabolism and sperm quality. In terms of testicular metabolism, the prediabetic group showed a reduction in the lactate content compared to the control group, and the ingestion of YT led to the maintenance of these lactate at normal levels. So the ingestion of YT was beneficial to the metabolism of YT of the testes. In terms of the epididymal metabolism, there was a general reduction of the glucose metabolism that we can see here in the figure, that many of the intermediates are reduced. And this resulted in a reduced lactate content. Although the ingestion of YT had less effect, it also reduced the lactate content. It was not able to reverse this reduction. In terms of the reproductive parameters, there was a decrease in the prediabetic animals drinking water, in the viability, and an increase in the percentage of abnormal of cells with abnormal morphology. And the animals drinking YT showed an increased motility regarding the other groups and a normal viability. So what we concluded was that prediabetes alters the testicular and epididymal metabolism, resulting in reduced sperm quality, and the YT consumption can prevent most of these metabolic alterations in the glucose metabolism induced by the disease, and improving mostly motility and viability. So the general conclusion of this project was that the regular consumption of YT can be a natural and cost-effective strategy to prevent prediabetes-induced reproductive dysfunctions, and the development of YT-based food supplements may enhance its therapeutic effect and allow its use in new antioxidant therapies for the treatment of male infertility and infertility. In fact, when I was in the United States, they were using some antioxidant therapies, but it was like trying and see the results, and then we change the dose. They don't know the amount of antioxidants they need to give to the patient. This project also highlighted that the beneficial effects of YT are not only due to one of its constituents, but rather to the combined effect of all its components. So this was a brief overview of my work during the PhD, and here I have some pictures of my previous research group. Luckily, these were times when we could socialize. And then in April 2020, I started working here at CISECO, at the University of Beira Interior, and unfortunately, in these new online terms, and mostly this is how I could meet many of you. So I started working from home, collaborating in writing projects and preparation of papers. Then in September, I could come to Aveiro, and I started learning some extraction and purification methods of several natural pigments. And I also applied for the Emporia Kitty 4KT Blue Economy Technology Transfer Program. This program, I was selected for this program, and this just started a few weeks ago, and it aims to leverage the skills and competencies of academic researchers to produce innovation and knowledge transfer within the blue economy sectors. So 12 researchers were selected per country among 5 participating countries, and we will work in early stage technologies to understand how to make them attractive for industries and pass from academia to the market. This is the consortium of this project, and it includes 14 partners in 5 Atlantic area countries. This year, I am starting new projects, and I prepared some research projects for degree students. I will be the supervisor of one project. I also am preparing projects for the FCT funding. And regarding the laboratory work, I am testing zwitter ions and natural pigments biocompatibility in different cell types. And this is like the link between what I was doing before and the work I am doing here now, because I also did many cytotoxicity assays before during my PhD. And I already have some preliminary results, but I will show you later in a future presentation. Thank you.
Thank you, Tânia. Thank you very much.